Hello, ma'am. Good evening, ma'am.
You can start, sir.
Yes, ma'am. Thank you. Good evening, my dear students. Last class, what we have discussed already in our about. So, what are the things in our about? See that plant breeding for the developing resistant to the insects, and crop varieties resistant to diseases, and then some important control measures for the plant diseases caused by the fungi, bacteria, virus, and nematode. Okay, and then plant diseases caused by the fungi, bacteria, and also virus, and protozoa, nematode, and so on. So, isn't it, my dear? Today, we are going to discuss about in this session the organic farming. Okay, the organic farming is also very, very important for the plant breeding and also the hybridization process. Here, see it, the organic farming. It is a method of farming. Okay, it's already in our but organic farming is already in our but there are so many farmers that were used organic farming. Okay, so then only they developed their agriculture process as well as the progress also, which is primarily aimed at cultivation land and raising crop in such a way as to keep the soil alive and the good healthy. That is very, very organic farming. What is the main aim of the organic farming? You know, two things is very, very important. So first one is considered here. Which one you know? See here, I will show you. Okay, soil improvement. Okay, my dear, soil improvement. And the next one is, what is that, you know, and also healthy in nature. How it will be healthy. Okay, so it is, these two things is very, very important, my dear. Okay, so for example, you know, but why we are, what is that? Changing the plants. Okay, because once we are planting the sugarcane, and another time chilies, and another times jam, maize, or onion, whatever it may be, we are rotating the plant cultivation also, isn't it? It is also one of the method which what are the nutrients not utilized by the previous cultivation, the plants also used. That is also another type of. So the soil is alive in nature. They keep the soil alive. How it will be alive in nature? How it will be alive in nature? That is very, very important. Okay, and in good health by use of organic waste. Organic waste means, you know, the organisms. Okay, organism means organic waste. What are the organic waste? Already you know about. So all the crabs, take an example, so animal and farm waste and aquatic waste, these are all considered as organic waste. It is also comes from the organisms. Okay, organisms, isn't it, my dear? Okay. And other biological material along with the beneficial microbes, biofertilizer is nothing but the microorganism used as a fertilizer to release the nutrient to crop for increase the sustainable production and eco-friendly. That is very, very important. Eco-friendly plant as well as the soil as well as the human beings also, and also microorganism. All are beneficial aspects only that were used. Such type of fertilizer is called as biofertilizers. There are so many biofertilizers there. Rhizobium, Azospirillum, Pseudomonas. Okay, all those things, they are all interrelated with the organic farming. And also, it is also used as eco-friendly. And at the same time, pollution, take examples of pollution-free environment. Suppose when you are using the urea, just to take an example. Okay, when you are using urea, what happens? It is also absorbed by the plant. Okay, plant, it will not take the taken by the plant directly. So the urea is converted into, see that formula is CHN, isn't it? Double bond CHN. Okay. The urea is converted into carbon, hydrogen is there. So the hydrogen is also split and also carbon is also split. Okay. CHN, N2. Okay. C double bond N, like that. CN2 double bond CN2, like that it will comes. Okay, the urea structure is that. The nitrogen is utilized by the plant with the help of the microorganism. What are the microorganisms present in it? Already you know about ammonifying bacteria, denatifying bacteria, and also nitrifying bacteria. So isn't it, my dear? See that. What are the characteristics of organic farming? First of all, you should know here. You should know two things is very, very important. One is known as the organic farming. What is the main uses? First of all, you should note it down. Keep the soil alive and good health. Okay, so that is very, very. What are the waste they were used? You know, only the organic waste only we are using. That's why the plants are healthy in nature and also they will not produce any diseases. That is also very, very important. And also biofertilizer, microorganisms, other living organism used as a fertilizer. It will enhance the rate of reaction. That means enhance the rate of production also. Okay, the yield also. That is also very, very important. How it will be? What are the characteristics of organic farming? See that protecting the long-term fertility. That is very, very important, my dear students. Of soil by maintaining organic matter level. This is the first point. Organic matter level. Providing crop nutrients indirectly using relatively insoluble nutrient sources. What is the meaning of insoluble? You know, some of the nutrition, it's not dissolved. They are all insoluble nature present in it. That insoluble nature is also converted into soluble nature with the presence of the microorganisms in the soil. It is also done. Okay. One more is the phosphate solubilization. Phosphate solubilization. Phosphate is not taken by the plant directly. Okay. So the phosphate solubilization bacteria is also present in the soil. It may convert into insoluble nutrient into soluble nutrients. Okay. And the next one is called as nitrogen. Nitrogen self-sufficient through the use of leguminous plants. Already you know all the leguminous plants, they stored nitrogen in the form of proteins. Maximum pulses, already you know about. Okay. The pulses, they contain more amount of nutrients, particularly the protein molecule, biomolecule, such as a biomolecule of which one, you know, protein is that. Okay, they need nitrogen for the formation of amino acid, and amino acids united together form as a protein molecules. That was done by which one, you know, the nitrogen's cycles. Already we studied in biogeocycles, isn't it? Biological nitrogen fixation. And it has a specific microorganism, ammonification, denitrification, nitrification, and also nitrogen fixation, all those things. Okay, already we studied in the biogeocycle, isn't it? And this is a third one. And the fourth one, weed, disease, and pest control relying primarily on crop rotations. Already I told you, the first time I told you, the crop rotation, why we are using this one, you know. Suppose when you are planting the bananas, what happened? You know, automatically after the complete harvesting, what happened? Completely destroy the plant, isn't it? And also they burn it. Why? Some of the microorganisms present in it, they can affect the new cultivation, new farming, like that. Okay. So and also natural predators, diversity, organic manure, and resistant variety. These are the important points. So that's why we are using organic farming. Okay, my dear. So you should note it down. This point is very, very important. And the next important point is what is meant by sustainable agriculture. Sustain. Okay. So what is it meaning? It is a system of agriculture that will last. Already you know, but sustain means affect. Which one you know? Suppose agriculture, how it will be affected? It maintain its productivity over the long run. Okay. That is very, very important. Take an example, 1950, one of the rice were grown. Like imagine. After 2025 also, the plant, it will be grow. How it will be? Suppose the production is, see here, already you know about it. So 1950, a particular variety of rice, IR8. Okay. So this plant, they will grow at the time 1 acre. How much you know? 500 bags, like that, imagine. Okay. 1950. 2000, just imagine. So this is 250 bags. They make 250 bags. 2025 also, you have to maintain 250 up to maybe 350, like that, bags. Okay, major bags. So here the increasing the order only, isn't it? It increases. Suppose if it is reverse manner, suppose the 1950, so it may be 250, and it gradually decreases 2025, 2020 or 25. What happened now? Only 100 bags only, it is a reversible manner, isn't it? That is the things they have given here. It is a system of agriculture that will last. It maintains its productivity over the long run. That is very, very important. Sustainable agriculture system are designed to take the maximum advantage of existing soil nutrients. That is very well. Existing soil nutrient and water cycle, energy flow, and soil organisms for food production. This is very, very important. These are the factors enhancing the desired the agriculture. Okay, suppose if it is sustainable, the process is very lost, like that, because these are the parameters are involved in this process. These are all these parameters. It will be very good in nature. The agriculture, it will not easily sustain. Okay, that is very, very important. Okay, my dear. Go to the next subheading is petrocorps. Okay, that is called as petroleum plants. The plant that produce latex containing liquids hydrocarbon. Already but take an example, papaya. Okay, and also another one is called as, what is that, Brassica family, rubber family, isn't it? Rubber. Okay, heavy lenses, like that, rubber family. They are all having which one, latex vessels, isn't it? Latex, milk secreting glands, secreting glands, isn't it, my dear? Secreting glands present in it. The white color liquid comes out from the plant body. Okay, latex vessel is there. It is also one of the special type of tissues present in the plants. They can use for some other purpose. For example, rubber is also used for the so many preparation is the tire and so on. So likewise, some of the plant, they have the latex containing liquid contain hydrocarbon. Hydrocarbon mostly used for the, and also is also used as a fuel, that are potential substitute for the petroleum. Example, curcuma langa. Already but sorry, curcumus, Euphorbia thirkalli. Just take an example. This is one of the plant. It is also otherwise called as a penicillin tree and also a gasoline tree. The latex can be converted into gasoline or oil. This is very, very important. For example, already resinous communis. Resinous communis. Resinous communis. Okay, my dears. Resinous communis is nothing but the which one, which soil, Okay, resinous communis is also one of the oil. Okay, that is called as biofuel. Biofuel, because the plant produce the oil. Okay, coconut is also biofuel. So sometimes they may use, take an examples. Euphorbia thirkalli. Don't forget, sometimes they may ask the question in these points. Which one of the plant it has a hydrocarbon, otherwise called as petroleum latex or gasoline, like that they may ask the question. This is also very, very important, my dear. Okay, go to the next one. Jatropha carcass. This is also another important. Jatropha carcass. I told you, Jatropha carcass. Kattamanak. Tamil we may call it as kattamanak. Okay. Seed oils used as a biofuel. Already I told you, this is very, very important. So many times they were asked the question this point. The last one is called as Puncomia pinnata, otherwise called as pungam tree. Okay, Puncomia pinnata, Carange, Panikragi. Okay, these are the another type of a plant. It's a small tree-like structure, Puncomia pinnata. Okay, it is a nitrogen fixation legume tree. It is also one of the leguminous trees. Already but seed oil is used as a lamp oil for making soap making also. Sometimes it may be used as a lubricant and also biodiesel. Don't forget, my dear students. Pungamia pinnata, Jatropha curcus. Okay. So these are the important, what is that? Hydrocarbon containing plant. Okay. So this is also very, very important, my dear students. NEET. Okay. Don't forget, my dear, Pungamia pinnata. It is also one of the nitrogen plant. Take an examples. The white color flowers also produce the leaves. Okay. Already you know, but the leaves look like this one. But there are some small, what is that? Protrude-like structure is there. Protrude-like structure. There is a small beads-like structure present in the leaves. The inside the leaves, what happened? There are so many microorganisms. Is there so many microorganisms present in it? Okay, my dear. So many microorganisms present in it. So this is also an important point. Next one is energy cropping. What is the meaning of energy cropping? It produces energy. Okay, how it will be? The energy, nothing but here you must mean it here, heat. Okay, major heat. How it will be produced? Heat. The rising crop for the production of ethanol. Ethanol is a source of which one you know. It is also used as a biofuel. Okay, ethanol. These crops are fermented to produce ethanol. How it will be fermented? You know about the sugar cane. Sugar cane waste. And also addition with the yeast. Okay. Here what happens? Sugar cane, it contains sucrose. Isn't it? Sucrose converted into ethanol plus carbon dioxide. So the ethanol used as a, which one you know? Fuel. Which is mixed with the petrol to form a gasol. Just note it down. Gasoline plus alcohol. You will get it. Which one you know? Gasol. See here. So already you know, but there are two parties there. Ethanol, HOL, isn't it? Here, petrol. Gasoline. Okay. Example, sugar cane. Already I told you. Sugar cane is also produced ethanol and potato ethanol, sugar beet ethanol, topika ethanol, maize ethanol. Like that, there are so many examples have given for the energy cropping plant. Okay, media. Here, you must note it down. What is meant by gasol? Like that, you know, sometimes they may ask the questions, the combination of ethanol with petrol. It is considered as a, it is named as a, what is a gasol? Okay, media. This is also another important NEET point of view. Okay, go to the next one. See that single cell protein. It is very, very important, my dear students. Take an example. All the questions, the single cell protein, SCP, must present 1000 times is important. 1000 times important in NEET question paper. Okay. Single cell protein. It is one of the algae. Okay. Single cell algae. There are so many examples has given. Chlorella. Okay. Chlorella, spirulina. So many things is there. But it contains highly rich, only one type of protein molecules, only one type of protein molecule, and also the amino acids, amino acids. A particular amino acids only present. Okay. Amino acids only present in the protein. Okay, see here, the conventional agriculture production of the cereals, pulses, vegetable, fruits, etc. may not be able to meet the demand of food for the increasing population. Suppose the increasing the population, automatically the food production is also increases. We have to increase. Then only they will get the food. Suppose if it is not correct, you will not get it. What happened? No, it will lack. Due to lack, what happened? So many diseases also caused it. Okay, media. The shift from grain to meat diet, okay, is also problem, since it's required 3 to 10 kg of grains to produce 1 kg of meat by animal farming. See here, so 3 kg to 10 kg, it is equal to 1 kg of meat. More than 25% of the human is suffering from the hunger and also malnutrition. This is already in our book. Okay, so take an example, Somalia. So many countries, what happened, you know, the poverty layer is also high. Okay, they will not get even single bread also. But what happened? They are having so much of money, but they will not get which one, food. Okay, so as a result, what happened? One day, 2 days, 3 days, all they will not get, means what happened? Finally, the nutrition deficiency also occurs. As a result, what happened? It will be suffering. Okay, some diseases or sick. Finally, lead to what happened? Already you know. So likewise, the second one is alternative source of proteins for animals and human. This is the very, very important alternative source of proteins. Some of the person, they will not take non-veg, meat, egg. Okay. The plant foods also, plant protein molecules also, sometimes they will not digest easily. Sometimes some of the person, they are having such type of capability. In such conditions, what they will do, they will alter the food sources. Which one, that's why they will choose a single cell protein. The single cell protein is protein rich biomass. First of all, you should note it down, which is used as a food or feed. Sometimes they may ask the question in China and Japanese people, they were used, which one, the spirulina tablet and spirulina food, and also chlorella, all those things used as a cattle feed. Sometimes it is also fashionable drugs, syrups, and also tablets like that, they were used as a SCP protein, single cell protein. The term single cell protein, however, is misleading because it is not only obtained from the unicellular microorganism, but also from the multicellular microorganism. It is not from the unicellular microorganism, but it is also multicellular organism. Take an example. The single cell protein is also made up of unicellular microorganism, multicellular microorganism, none of these like that. So don't forget, the single cell protein is also made up of multicellular microorganism. From a nutritional point of view, the nucleic acid content in SCP is one of the main factors hindering its utilization as a food. Okay, and the excessive intake of nucleic acid purine in human diet result in uric acid precipitation. Already you know a little bit, when you are taking more amount of protein molecule, it can be easily deposited in the your kidney or blood or liver. So as a result, what happened? No, the protein molecule converted into urea or uric acid and deposited the blood, and also it will transfer through the blood circulation. Finally, what happened? It will lead to a toxic effect. Okay, the toxic, when the uric acid deposited in your blood, what happened? Automatically your joint pains and gout and kidney stone formation takes place. So their nucleic acid content, okay, must be reduced below 2%. How much is the 2%? That is also very, very important. Those who are having proteinuria, protein excreted in the urine, that condition is called as proteinuria. Due to the, what is the reabsorption of protein is lacked in the kidney molecule, kidney. As a result, the molecule it will not easily absorbed, reabsorbed, resorption. So what happened? The protein molecule converted to purine component. That is very, very important. The nucleic acid, particularly purine. What are the purine? Is the already adenine and also guanine. These are the two components essential for the production of uric acid. It is deposited in the kidney or gout. It will lead to some difficulties. Okay, my dears, giddiness and joint pains and stone formation in the kidney and stomach pain, kidney and the abdomen pain, like that it will be created. Okay, see here. The single cell protein is rich in good quantity of protein and poor in fat. This is the main aim, main content. What is the main content of this one? Suppose if you are taking the single cell protein, okay, foods, a good quality of protein, but poor in fat. That's why most of the people took this one. They will take. High protein and low fat are desirable features of human food. That's the reason. However, care has to be taken to remove excess of nucleic acid and toxin present in the biomass. The raw material for the single cell protein productions included waste sulfide waste liquors. Whey sulfate. Whey means what is that? After the curd preparation, there is a yellow color fluid is there, isn't it? Whey sulfate. Here they may be differentiated. Whey sulfite waste liquor, straw molasses, and animal manure. These are the important. The microorganisms are growing in a large fermentation tank. That's just you note it down, my dear students. The fermentation tank is there with forced aeration for vigorous cell growth. Vigorous cell growth, rapid growth is there. Incidentally, such utilization also reduce environmental pollution. The microorganisms which can be explored for obtained a single cell protein are as follows. This is very, very important. Just you should note it down. Two things is very, very important. Particularly, the single cell protein is highly rich in protein molecule, and second one is less fat molecules. First of all, second molecules. What is that? Second point is that what is that? The removal of excess of nucleic acid and toxin present in the biomass. Second point. At the same time, the growth of the lab or cultivation of the single cell protein is also very rapid, where they were used the fermenting tank. Fermenting tank already know 1,000 gallon, 10,000 gallon, 50,000 gallon is like that, isn't it? It's a large tank. And at the same time, aeration is also very, very important. So the aerobic condition, presence of oxygen is very, very important. Light is also very, very important for the production of single cell protein molecules. The microorganisms, what are the microorganisms which can be explored for obtaining single cell protein are as the follows. Sometimes they may ask the question, what are the microorganisms used as which one for making single cell protein cultivation or making process or in ferment or like that, they may ask the question. First of all, is not a fungi. Which fungi they will use? The Fusarium graminarium. Fusarium graminarium. It's a new one, isn't it? Already you know about yeast. Sometimes they may ask the question, yeast zoological name. Sometimes it may be a biological name. So botany, isn't it? Okay, here the botany is the correct one. Candida utilis. Candida utilis is the zoological name. Oh, sorry, botany name. Because it's a fungi, isn't it, my dear? So unicellular fungi, except unicellular fungi. Which one Yeast is a unicellular fungi. Others are multicellular. And also the mushrooms, already you know, but the mushroom structure look like this one, isn't it? There are so many mushrooms is also there. Okay. These are all are considered fungi. And cyanobacteria, don't miss it, my dear students. Spirulina. Spirulina is one of the, what is that? Cyanobacteria, just like I said, Nostoc anabina, like that, cyanobacteria. And green algae, this is very, very important. Most of the cases, they ask the question, green algae, chlorella, and also a Schinidismus. Okay. And bacteria, Methylophilus, Methylotropous. Methyl means innovable, CH3, isn't it, my dear? So these are the bacteria. So you have to note it down. The 4 microorganisms involved in this process. First of all, you should note it down. Fungi is the first one. And also second one is cyanobacteria and algae and bacteria. Particularly, this is very, very important. Methophilus, Methylotropous. These are the 2 bacteria also involved in this process. Methophilus, a source of protein, 250 grams of this microorganism can be, which one expected to produce 25 tons of protein per day. Imagine Methophilus, source of, which one 250 grams, how much it will be produced? 25 tons of proteins they will be producing per day. See there, 250 grams, 25 tons. Imagine the huge amount is there. My dear students, don't forget here. Next, we will pass on to the next one is question part. The first part is finished. They evaluate yourselves. The Nobel laureate who developed semi-dwarf what varieties, otherwise called as wheat varieties, in Mexico was, who is that? Here, any Green Revolution process is the Green Revolution scientist is the Norman Borlaug, correct? Isn't it? He is the worldwide, okay? Herbert Boyer, no. Williams Harvey, no. Typhoid Mary, no. Who is the correct one? See this one? First one is the correct one. Okay. First one is the Nobel laureate who developed it. So Nobel laureate means Nobel Prize. Who got it? Nobel Prize for the Green Revolution. And particularly semi-dwarf variety was discovered by the scientist there. The Mexico was the Norman E. Porlock. First one is the correct one. Second one is Sonalika and Kalyana. Sona are the variety of which one you know? So sugar cane or millets or wheat or rice. Which one is the correct answer? Just to imagine. So wheat is the already we discussed. Okay. Sonalika, Kalyan. So these are the important points of wheat. Jaya and Ratna are the semi-dwarf variety of which one you know? Wheat, cowpea, rice or mustard. Which one is the correct one? See this one? Second one is the correct one. Pusa, Komal, variety of cowpea is resistant to the disease of bacterial flight. Okay, so already you saw it that the answer is also there. So already you know about. So which one is the correct one? This one is the correct one. The bill bunt is not comes, white rust is will not comes, and the leaf curl is not done. Which one you know? Pusa, Komal, the cowpea. So that is also one of the bacterial blight. Triticale, just already we discussed it. Which one you know? Triticale, kale, sea kale. Which is the correct answer, you know? Wheat and rye only. See that one answer? Okay, see that one? First the answer is there. First one is the correct answer. Isn't it, my dear? Wheat and rye. Wheat means triticale, canadensis, isn't it? Like that you studied it. Rye means sickle, like that. Company will get triticale. So this is the triticale. So combination of the two species. Okay. As a one. So as a result, you will get it the single. So this is the correct one. Okay, this is the correct one. Okay, my dear, see that one. So that's all about the first part. We will pass on to the next one, my dear students. Second part. See here. So this is very, very important, my dear. This is also second part, continuity of the. So first of all, you should note it down. Before that, what is meant by tissue culture? What is meant by tissue culture? Already you know about tissue culture is nothing but one of the technology they were used for the what is that? They were used for the laboratory aspects. What is meant by tissue culture? Okay, Media. Tissue culture. What is the main uses of the tissue culture? Will you please tell me what is by? Okay. Okay, tissue culture. First of all, you note it down. Any one of the plant part. Take an example of any one of the plant part. Whether it may be a root or shoot. Okay. And flower, okay, flower or anther, calyx, corolla, whatever it may be, okay, whatever it may be. It can grow into what is that? In the test tubes, grow into the small test tubes in vitro in nature, in the laboratory aspects, okay. What is that? This is agar medium. Otherwise called as what is that? Nutritive medium. The plant we can cut any part of the plant body. Already I told you, any part. Just take an example, any one of the tip, cut into place here. Okay. Placed here inside. Okay. So what happened this one? Any part of the plant body, I told you, this is any part. Okay. It gives the entire plant is called as what is that, you know, tissue culture. Entire plant is called as tissue culture. Okay, this is also one of the methodology. For example, it is also absorb the nutrition present in the agar medium. Okay, it will grow and also undergo the mitotic cell division. Mitotic. What is the main process of mitotic? Single cell converted into double, isn't it? double again converted into tetra. Tetra into octa. Okay, 8. So single cell, double cell, 4, 8, and 16, 32, like that. So finally, what happened? Oh, the colorless mass of cells also produced. The colorless mass of cell is produced within the test tubes. That is called as callus, undifferentiated. Okay, callus cell is there. After the callus cells also undergo the subculture. Okay, subculture. You will get it. Which one? You know, pure culture, isn't it? Subculture. What some of the deep impurities also removed through this process. So likewise, these are the processes. Here we get the culture under the laboratory conditions from the plant directly we have taken, isn't it? In this case, what can it produce the callus? Again, the callus is also undergo the subculture. So okay, subculture. Okay, here the culture is also again. You have to practice more than 10 times. What happened? No, this is the first test tube, second, third, fourth, fifth, sixth, seventh, eighth, 9, and 10, like that. Imagine. Okay. So here too, again, the first test tube, second test tube, you have to transfer like this. Now, finally, the 10th one. So 10th one, it will be good in nature. That 10th one, it will be a, how it will be? Very good. Callus formation is there. After that, what happened? No, you have to add some axin, cytokinin, and the phytohormones is there. After that, addition of cytokinin is already present in it, but at the same time, what happened? The callus undergoes the growth of the plantlets within the test tubes. What happened? There are so many plantlets have been produced. The plantlets also transferred to the land. Okay, plantlets also transferred to the land. That process is called as gardening. You must note it down here. The any part of the plant body, it gives a entire plant. That ability, that potential is called as totipotency. What is that? Totipotency. That is very, very important. That things we are going to discuss in this part, my dear students. So first of all, you note it down. Very, very important. Okay, see here. What is meant by tissue culture? What is meant by tissue culture? It was found by the scientists during the 1950s. 1950s. That whole plants could be regenerated. That is very, very important. From any part of the plant, take out and grow in test tubes under sterile conditions. In special nutrient media. What is meaning a special nutrient media? The special nutrition media is nothing but the agar. The agar is also isolated from the one of the algae. What is that? Brown algae. Gelidium. Okay, all those things already we know all about. The capacity to generate a whole plant from any cell is called as totipotent. That's why I told you. What is by totipotency? Sometimes they may ask the questions. What is by totipotency? Which one of the following statement is not correct with the regarding totipotency? This capability or capacity to generate a whole plant from any single cell, just imagine single cell, is called as totipotency. In commercial setting, tissue culture is often referred to as the micropropagation. Once again, they ask the question, sir. Totipotency also called as micropropagation, and one more is the vegetative propagation, like that. It is also one of the mitotic cell division, vegetative propagation, micropropagation, and then totipotency. These are the 4 categories they may ask the questions, my dear. So you have to ready. You should ready for these questions, which is in fact one of the technique is in tissue culture. Tissue culture. What is the meaning of tissue culture? You know all about undergo the meristematic tissue. Each and every plant, they are having the meristematic tissues. Okay. Which one it undergoes the cell division process, you know? Okay. So only the meristematic story. Meristem. Isn't it, my dear? Meristematic tissue only. So the plant having meristemic continuous cell division, that is totipotency characteristics. Isn't it, my dear? Okay. See, this is the name of the scientist, the Haberland. Don't forget, my dear students, Haberland. What is the name of the scientist? No, Haber. See, Haberland D. Okay. Haberland is the scientist who was the first scientist to produce a whole plant from plant tissue, and see, he is popularly called as father of tissue culture. Who is the father of tissue culture? Haberland. Don't forget, this is very, very important, my dear students. Here you must note it down. 3 point is very, very important. First of all, what is my tissue culture? What is my totipotency? What is my vegetative propagation? Otherwise called as micro propagation. Okay. Who is the father of tissue culture? Okay. So that is very, very important, my dear student. Don't forget, keep on practice. The plant tissue cultures, just imagine how it will be. Already I showed it. Here, the plant tissue culture is the culture and maintenance of the plant cells or organic in sterile, nutritionally, and environmentally supportive conditions. That is called as in vitro. In vitro means nothing but the lab. Okay. In vivo, it's nothing but the outside. Okay, outside the lab. The plant tissue culture consists of the pollen tube, pollen culture, callus culture, callus tip culture, stem tip culture, meristem tip culture, and protoplasm fusion. These are the techniques that were used. These are the plant also, they can grow under the lab or in vitro. Okay, media. Okay, media. So this is very, very plant tissue culture consists of pollen culture. What is the meaning of pollen culture? Already know pollen grain is nothing but the micro, isn't it? Microspore. Anther. Callus already we discussed. Stem tip culture, meristem tip culture, and also protoplasmic fusions. These are all comes in the anatomical, isn't it? Stem, meristem, all those things. The plant tissue culture are often classified according to type of in vitro growth, namely callus, suspension culture. What is my callus means? It is a solid nature. Suspension is nothing but the solution format. Okay, suspension. Explant used for the culture initiation, for example, embryo culture, anther culture. These are the another type. So these are the two types of plant tissue culture according to their nature. Type of in vitro growth, in vitro inside the lab only, namely callus. Callus only already told you within the test tubes only in the aseptic conditions, isn't it? Have to put into the oven. Okay, there is a lab chamber is there, small chamber like that. The boxes look like this one. There is a two hole is there. Okay, so the chamber look like this one. You have to insert your two hand inside it, and also inside there are so many culture. Mother culture is also there. The lamp is also there, and also all are in sterile condition. The upper side there is a UV light is also, tube light is also there present in it for the what is that sterilization process. Okay, then an explant is a plant pod that is exist from its original location and used for the initiation of culture. So this is the process. See, tissue culture offers significant benefits over traditional propagation method. Significant. What are the main significance of the tissue culture method? This is also sometimes they ask the question. Much faster rate of growth. This is very, very important. Much faster. When it will be possible, seedless plant. Some of the seedless plant is there. Just imagine, seedless plant is there. The seedless plants, they will produce which one, they are all commercial oriented. Isn't it? Take an examples of what is it? Sapota, banana, they do not have the seeds, but the sapota, they are having the seeds, but they will not grow. Only for the grafting method and also cutting, grafting, layering, that method they were used. At the same time, we can not get, suppose we are planting this plant by seeds, it will take some more times. Take an example, 5 years, 6 years, 7 years. Suppose you are using this method, you will get the result within short period. That's why they have chosen tissue culture method. So it is a very, very important, much faster rate in rate of growth. Production of large number of genetically identical clone. I need a single plant in the, for example, take an example, Tectona grandis teak or sandal plant is there. Okay, sandal plants also I need a 1,000 millions like that in a single room. How it will be possible? Why only in this method only we can be possible within short time. That is very, very important. Within short time, how many plantlets we can produce? 1 lakh plantlets. You can put 10 lakh, 1 lakh like that within the lab. Particularly a single test tube is enough for the production of this much of plantlets. Okay, within short period, within a month, or within 10 days or 24 days. Okay, like that. Maximum it will take 1 month. We will get it. How much plantlets? This much of plantlets. Okay, media. Tissue culture technique are used for the virus eradication, genetic manipulation, somatic hybridization. These are the 3 important points. Why we are using, virus eradications. Suppose some of the plants affected by the virus, it cannot be able to produce again newly, means that time they were used tissue culture methodology. Genetic manipulation, for example, mixing of 2 genes by the process called as, which one genetic engineering, biotechnology, as well as protoplasm. Somatic hybridization. Somatic is also one of the protoplasm fusion is also mitotic cell. Somatic cell is also called as mitotic cells also united together. So take an example, A and B. The cell is there. Okay, the cell is there. Okay, it has the extra chromosome body plasmids. Here also there is an extra chromosome body. Both are united together, form a new variety. Isn't it, my dear? So these are the somatic hybridizations. So the somatic hybridization, sometimes they may ask the questions, which one? The tissue culture is also one of the somatic hybridization. Which one of the following technique is possible like that? Virus eradication, genetic manipulation, somatic hybridization, and also meiotic cell division is also occur like that. Meiotic cell division is not occur. You have to find out the correct answer. That is very, very important, my student. See, this is the method. So this is the important points, my dear. Okay. So here a carrot have taken. After that, you have to made a cut in cross section, transverse sections of the carrot root. Again, it will be okay, cut into a 2 mg fragments. Simple one. It can be introduced into the which one culture medium. The fragments were cultured in the nutrient medium. Stirring caused single cells to soak off into the liquid. So this one. After that, single cell free in suspension began to divide. So here, what happened? The single cell divided into 2. Again, the embryonic axis also developed. The plantlets was also cultured. After that, you will get the entire plant. So here we are using only the single cell from the parent plant, but we are getting the exactly similar to the parent. So this process is called, it is also a totipotency. Single cell developed into the entire plant. That condition is called as totipotency. This is the method. This is the method, my dear students. Okay. So go to the next one. See that. Media composition. Very, very important, my dear student. This is very, very important. So many times they were asked the questions. Media composition, particularly MS medium. The MS, otherwise called as mosaic and hook medium. First of all, is not MS medium. What is the meaning of MS? See here. Mura hits and hook medium. Which year? 1962. There are so many medium is there. MS medium is there. White medium is there. So many medium is there. What is the difference between these medium? The DTT substances, the amount it will be differ. Vitamins, mineral, phytohormones. That things also differ. It is also widely used in different culture system and generally consists of what are the MS medium content. Sometimes they may ask the pH is very, very important. And also, what about the temperature? Which temperature it undergoes the process? That is 24 degrees Celsius. This is the temperature, isn't it? The pH, it will be 5 to 6. 5 to 6. That is nothing but the acid pH. isn't it? Don't forget, my dear student. These are the two things is very, very important. See, your main, what are the composition is important for the growth of the tissue culture. Carbon sources is very, very important. Okay. And also nitrogen source is also very, very important. Plant hormone is also very, very important. Okay. See there. The carbon and the energy sources such as sucrose and glucose. Sometimes they ask the fructose, lactose, galactose, maltose, like that they may ask the questions. Particularly, these are the two carbon sources is very easily, what is that? Utilized by the plant. That's why they have chosen glucose and sucrose. Vitamins such as vitamin B1, thiamine, nicotinic acid, okay, pyridoxine. See that vitamin B6, pyridoxines, nicotinamides, B5, B1, B5, B6. These are the three vitamins is essential for the, which one MS Cook medium. And then organic nitrogen from the amino acids such as glutamine and also asparagine. These are the two amino acids also very, very important for the tissue culture methodology. That means what is that? That agar medium, that MS Cook medium is containing, these are the components. These are the components support to the growth of the plantlets or plant or tissue culture or culture, callus, whatever it may be. Particularly the growth regulators is otherwise called as what is phytoregulator or phytohormones. What are the phytohormones is very, very important. Axin is also very, very important because it is also essential for the growth, cell division and elongation, like that. 2,4-D. Okay, 2,4-D. Cytokinin, cell division. It's very, very important. BAP, gelatin and gibberellin. These are the three things. Axin, cytokinin, and gelatin, gibberellin. These are the phytohormones essential for the growth are present in the MS medium. A variety of extract, namely protein hydrolase, yeast extract, malt extract, coconut milk, orange, and tomato. So these are the things. What is that? The yeast, hydrolase. Take an example. So coconut milk. Okay. The cytokinin is mostly present in it. Cytokinin is important things. Coconut milk. Okay. So these are the components mostly present in the where the MS medium, otherwise called as Murasek and Cook medium. Carbon source, vitamins, organic nitrogen, growth regulator, and extract is also very, very important. Last one is called as gelling agent. Otherwise called as gelling agent. Simply called as gelling agent. Gelling means mixing. Combinations such as agar-agar, gel-like substances. Gelidium, already told you. It's a liquid, semi-solid liquid. Okay, semi-solid nature. Okay. So then a plant, it will be absorbed. Okay. So these are the mixtures very, very essential, and also it kept about how much degree Celsius? 24 degrees Celsius with some illuminations. Nutrition pH range from the 5 degrees Celsius, 5 to 6 for the suitable aerations. Okay, in liquid medium culture, air is provided through the filter paper, bridge, or soaking, or passing of sterilized air. So these are the important things. How will you make agar? You have to prepare the sterilized flask. Okay, so that thing is also very, very important. For example, take an example. Okay, see this one. So what about the flask? Isn't it, my dear? Flask is that. Just imagine this is considered a flask. Inside what happened, there is a agar medium is there. All the nutrition substance is also there. Agar medium, isn't it? Agar is a gelling agent. Inside what happened, nitrogen sources, carbon sources, phytohormones, isn't it? Hormones is also there. And also gelling agent, already told you, isn't it? And also what is that? Culture medium, cultures. Okay. So what about the temperature? Already 24 degrees Celsius. What about the pH? About 5 to 6, isn't it? So after that, what happened? It may be sterile conditions only, sterile conditions, completely closed with the cotton plug. Okay, cotton plug. These are the cotton plug. Okay, already you know about there is a explant is that. Just imagine there's a explant is also present in it. Okay, inside is there. Okay, what are the plant? Whatever it may be. Any plant, you can take an example sample. After that, you have to select the test tubes. Okay, test tubes, and also already in our board. Test tubes also you have to pour the agar. Okay, I will get it to the slanting position. Why we are putting in this like no? So for example, take a small beads like structure is there. You put a test tubes. You take a test tube and also pour the agar. Okay, agar solution. After that, you place that a slanting position like that. You have to place it like this slanting position. So that time what happened? No, you will get it like this. Why we are putting like this? No, the space is high compared to the normal test tubes. Just we are putting like this, no, straight forward. What you know, the space is very less in nature compared to this one. So here it will take more. Here it will be get. So for example, there is 3 centimeter. You know, here the 6 centimeter. So this one is possible. We can place it here to here. Okay, how many plantlets? Say we can place it 1 or 2, whatever it may be. We can place it inside the. What is that? Plantlets. So that is a very, very important. So here we are placing the plantlets from the, so this one aseptic condition only and kept under the light sources and so on. So then only what happened, the plantlets absorb the nutrition. Sorry, that yes, the plant meristematic tissues absorb the nutrition and also it will be grow. Okay, completely it will be grow as a plantlets within the lab only. After that, it will be passed on to the hardening process in the land process, lab to land. Okay, that is called as hardening. Actually, discuss my dear. Okay. See this one. See, and sterilization technique is also there. Sterilization, all the equipments you must sterilize. Sterilization is the procedure we used for the elimination of the microorganism from the all equipments what we are using for the tissue culture process. The maintenance of the aseptic free from the all microorganism or sterile condition is essential for the successful tissue culture procedure. Otherwise, what happens? No, the disease, the tissue culture is completely a flop. Why? Because microorganisms present everywhere in the soil as well as the air also. The fungus is also present and it can be easily enter because the growth medium it easily absorb the microorganism easily. The need for the asepsis requires that all culture vessels, media and instruments used to handling tissues as well as the explant itself be sterilized. How it will be sterilized? With the help of the alcohol. All the operations are carried out in laminar airflow chamber. That's a laminar airflow starlight cabinet. It's nothing but the airflow chamber. This is very, very important. So that's why I told you laminar chamber. The laminar chamber just like a wooden box. Okay, just like a box. Okay. There is a small 2 opening is there for your hand insertion. Okay. Inside what happened? There is a lamp is there. Inside there is a lamp is there. And so many stand is also there with the test tubes. Okay, all those things present in it. You have to put the, what is one, you know, you have to take the, what is the sterilization process with the help of the alcohol. Okay, you have to pour, your hand is also washed with the alcohol. Then only you enter the hand inside. You have to put inside hand. Okay. So under sterilized conditions, you are doing all those things. It is essential that the explant culture vessels, media, and the instruments used for the plant tissue culture be made free for microbes. That is the first point. The explants are therefore treated with the specific antimicrobial chemicals also, antimicrobial chemicals. Flame sterilization method is used for the sterilization. This is also another method, and the instruments that are continuously used during the manipulations work. Instruments are soaked in 70 to 80% of alcohol, already told you, followed by flaming on burner in the laminar airflow hoods. Okay, how it will be like that? Already told you, isn't it, my dears? Inside. The most nutrition media are sterilized by using the autoclave only. Autoclave, just imagine autoclave means it's nothing but the instruments that's like a cooker, but it's a very large vessels. The standard conditions of autoclave media is 21 degrees Celsius. Just imagine with a pressure 50 Pascal for 20 minutes. This is very, very important, my dears. When you are placing, take an examples that which one, autoclave. Just to take an example, so autoclave like this. There is a lid is also there. There is a whistle is also there. Okay, there is just like a pressure cooker. Okay, inside what are the things is you are going to place there? They take an example, test tubes. Okay, and there is a conical flask, whatever it may be. You have to place it and kept under the sterilization heating process. Inside there is a bath, water bath is also water. Take an example, small vessel is there. You have to place it here with the cotton, cover with the cotton, and paper also you have to tie with it within it. Okay, and then close it and also closed it. So then only what happened on the inner side already you have to wash with the alcohol and also chemical substances also you have to use it, particularly 0.1% HCl and nitric acid they were used. After that, what happened? No, alcohol they were used, and then wrapped with the paper and then placed inside the oven and closed it and kept for how many degrees Celsius? 121 degrees Celsius. How many minutes you have to take? 15 Pascal and 20 minutes. Okay, my dear. And plant material can be surface sterilized by the variety of chemical involved in the eradication of microorganism with the aid of chemicals such as solution sodium hypochlorite, calcium hypochlorite, and bromine water and mercury chloride. Maximum they were used to mercury chloride, 70% alcohol, hydrogen peroxide, etc. These are the chemicals they were used for the sterilization of plants. Okay, and callus culture is there, and how it will be subculture. So all those things we will see the remaining things on the next class, my dear students. Today we will finish it up to this level only because the time is not sufficient. So we'll see the remaining things on the next class, my dear. Okay. Okay. Thank you so much, ma'am.
Thank you, sir.